Hi, and welcome to today's video. Today we'll be discussing Simon's two-stage design. First, we'll give a summary of Simon's two-stage design. Simon's two-stage design is a statistical method commonly used in phase 2 clinical trials. This design helps determine whether a new treatment shows enough promise to warrant further investigation in larger phase 3 trials. The main advantage of this design is its efficiency in terms of sample size and ethical considerations, ensuring that fewer patients are exposed to potentially ineffective treatments. Where and why it is used. Simon's two-stage design is often employed in oncology and other medical fields where treatments need to be evaluated quickly and ethically. Researchers use this design to make preliminary assessments of a new treatment's efficacy while minimizing the number of patients needed. By structuring the trial in two stages, the design allows for early stopping if the treatment is clearly ineffective, thereby reducing unnecessary patient exposure to ineffective therapies and conserving resources. Typical sample size. The total sample size in a Simon's two-stage design is typically small, usually ranging from 20 to 50 patients. The exact number depends on several factors, including the hypothesized response rates and the acceptable levels of type 1 and type 2 errors. The sample size is split between two stages, with the first stage often including about half of the total number of patients. The parameters: P naught, null hypothesis response rate. The response rate under the null hypothesis representing the maximum response rate that would indicate the treatment is ineffective. P1, alternative hypothesis response rate. The response rate under the alternative hypothesis representing the minimum response rate that would indicate the treatment is effective. Alpha, type 1 error rate. The probability of falsely rejecting the null hypothesis when it is true, also known as the significance level. Beta, type 2 error rate. The probability of failing to reject the null hypothesis when the alternative hypothesis is true. The power of the test is 1 minus beta. Key design components. N1, first stage sample size. The number of patients enrolled and evaluated in the first stage of the trial. N, total sample size. The total number of patients enrolled and evaluated if the trial proceeds to the second stage. R1, first stage critical value. The maximum number of responses in the first stage that would result in stopping the trial for futility. R, overall critical value. The maximum number of responses required to consider the treatment effective after both stages are completed. Important statistics. The probability of early termination and expected sample size if the treatment is inefficient are given by the following formulas below. Type 1 and type 2 errors. Let B with parameters nxp denote the binomial distribution, and let capital B with parameters nxp denote the cumulative binomial distribution. For pre-specified response levels P0 and P1, the actual type 1 error rate alpha prime and the actual type 2 error rate beta prime can be calculated for any given parameter set R1, N1, R, and N in the following manner. Steps in Simon's two-stage design. First stage, enroll N1 patients and observe the number of responses R1. If the number of responses is less than or equal to R1, the trial is stopped for futility. If the number of responses is greater than R1, proceed to the second stage. Second stage, enroll the remaining N2 patients, observe the total number of responses R from both stages. If the total number of responses is greater than or equal to R, the treatment is considered effective. If the total number of responses is less than R, the treatment is considered ineffective. By using Simon's two-stage design, researchers can efficiently and ethically determine whether a new treatment has the potential to be beneficial while controlling the probabilities of type 1 and type 2 errors. Hi and welcome back. And now we'll be creating a Simon's two-stage design in SAS Studio to create a data frame that contains all of the trials that comply to a particular parameters that we pre-specify. So for the first line of code, we'll create a macro with the statement macro Simon's two-stage design with parameters user beta, P0, and P1. User n will be the user specified sample size. Alpha will be our type 1 error rate, will be our type 2 error rate. P0 will be the null hypothesis response rate, and P1 will be the alternative response rate. For the step, we'll create our data frame. We'll create a data frame df1 and write a loop. Do n1 equals 1 to user n minus 1. So in effect, we're creating a loop from our first stage sample size up to 1 minus the overall sample size. Next, we'll create a second loop where we loop from R1 equal to 0 up to N1 minus 1. Next, the output statement, and we'll end each loop. Great. Now, we're going to create a loop in the same data frame, looping from R1 plus 1 to the user specified place. This will be our response rate for the second stage. Next, we're going to specify the alpha and beta parameters that have been set into data frame 1. The probability of early termination under the null and alternative hypotheses. Next, we'll set the iterator for our summation sigma. This is used to calculate the summation of the products between the PDF, and the CDF functions. Next, we'll look at data frame 2. This is where we have the Simon's two-stage designs. Now we need to loop through the values of R. Loop setting R equals N2 plus 1 to R1 by minus 1. So in effect, we're iterating down from the second stage sample size plus the response in stage 1 down to the response rate in stage 1. Next, we initialize probability of early termination under the null and alternative hypotheses. We can cut our code from before and simplify that. Great. Now, we need to reset the sums each R value. We have to loop through the value of L. We'll loop from R1 plus 1 through the minimum N1 and R. Now, we need to set some criteria. Minus L is greater than 0, and if N2 is greater than R minus L, then we will do the following. Add P naught becomes the PDF of a binomial distribution with parameters L and 1 multiplied by the CDF of a binomial distribution with parameters R minus L, P naught, and N2. Similarly, for add P1, we will calculate the f of a binomial with parameters L, P1, and N1 by the CDF of a binomial distribution with parameters R minus L, P1. Next, we'll update our sum parameter and this is equal to the sum of add P naught plus add P naught. Sum of P1 is equal to the sum of add P1 plus add P1. And we'll close our first loop, second loop. Next, we'll calculate the P naught S and P1 are effectively our type 1 and type 2 error rates. Next, we have to check the conditions for Simon's two-stage design. So, if P naught is less than or equal to our alpha value, and P1S is greater than or equal to 1 minus, then do the following. Sub S, our user n value, and 1s is equal to n2s is equal to n2. R1s is equal to R1. Rs is equal to R. Probability of determination S is equal to PET naught. Likewise, expected sample size S equals N1 plus 1 minus probability of early termination under the null hypothesis times N2. Output end the first loop and the second loop. And so let's close our macro by using the mend function, Simon's two-stage design. Let's plan to run this with the following parameters. We will say Simon's two-stage design, 46, 0.1, 0.1 for both alpha and beta. Next, for our null hypothesis rate, we'll say 30% or 0.3, and the alternative response rate at 0.5. And on our code, and as we can see, we have 32,000 rows, 432. And there's quite a few designs. We'll have to obtain the minimax and optimal designs in the next video. So for now, I'll say so, and be prepared for the next video, which will discuss the minimax and optimal designs of a Simon's two-stage design.